Scientists find form of CRISPR gene editing with new capabilities. Just a few years ago, CRISPR was a cipher, something that sounded to most ears like a device for keeping lettuce fresh. Today, CRISPR-Cas9 is widely known as a powerful way to edit genes. Scientists are deploying it in promising experiments, and a number of companies are already using it to develop drugs to treat conditions ranging from cancer to sickle cell anemia. Yet there is still a lot of misunderstanding around it. CRISPR describes a series of DNA sequences discovered in microbes, part of a system to defend against attacking viruses. Microbes make thousands of forms of CRISPR, most of which are just starting to be investigated by scientists. If they can be harnessed, some may bring changes to medicine that we can barely imagine. On Thursday, in the journal Science, researchers demonstrated just how much is left to discover. They found that an ordinary mouth bacterium makes a form of CRISPR that breaks apart not DNA, but RNA, the molecular messenger used by cells to turn genes into proteins. If scientists can get this process to work in human cells, they may open up a new front in gene engineering, gaining the ability to precisely adjust the proteins in cells, for instance, or to target cancer cells. The groundbreaking thing about this work is that it now opens up the RNA world to CRISPR, said Oliver Rackham, a synthetic biologist at the University of Western Australia who was not involved in the study. CRISPR was first discovered in 1987, but it took decades for scientists to figure out that microbes needed the system to recognize DNA from invading viruses and to chop it into pieces, stopping the infection. In 2012, a team of scientists led by Jennifer Doudna of the University of California, Berkeley, and Emmanuel Charpentier, then at Umeå University in Sweden, discovered how to use this microbial defense as a gene editing tool that could potentially alter any piece of DNA.